Hello, let's discuss on the semi-conservative methods of DNA replications. DNA replication refers to the duplications of the DNA molecules to make the identical copy of itself. So we know that the nitrogenous base in the DNA or RNA are specific. That means A will always pair with T in case of DNA. In case of RNA, A will always pair with U with 2 hydrogen bond, and G, that is guanine, will always pair with cytosine with the 3 hydrogen bond. So this is pairing is universal. Now, according to this semi-conservative model, during DNA replications, the hydrogen bonds that are pairing the two nucleotide in the polynucleotide chains breaks down and gets separated, exposing its nitrogenous base. So these are the nitrogenous base that are paired in the two polynucleotide strands of the DNA. So let's say here A always pair with T, G pair with C. Now in this case A is pairing with T, G with C, A with T, T with A. Now the hydrogen during DNA replication, this hydrogen bond, since it is a weak bond, it breaks and it separates the two strands of the DNA, exposing its nitrogenous base. So one of the strands of the DNA will be acting as a template, other is just a complementary strand. We know that. Now once the strands of the DNA get separated due to base specific pairing, the separated strands exposing the nitrogenous base attract its own complementary nitrogenous base again. That means its own complementary strand. So this is the breaks or the exposing nitrogenous base. So it will attract its complementary strand carrying complementary DNA sequence. Complementary nitrogenous sequence. So if this strand is carrying C nitrogenous base, then the complementary strand that it will attract will be G. This G will attract C, A will attract T, T will attract A. So the complementary strand will be carrying the nitrogenous base, which will pair exactly with this template. Same as in this case, the complementary strand will be carrying the same nitrogenous base that will exactly pair with these DNA strands. So these keep on continuing, and the separated strands DNA will be attracting the complementary strands having the complementary nitrogenous base. So this will keep on continuing till a new polynucleotide chain. That means if this is the separated DNA, a new polynucleotide chain, this is the new complementary strand, this will be the complementary strand that is running in this side and running this side. So this will keep on continuing till a new DNA strand is formed again. So from this it is clear that the polynucleotide strands of the DNA helix serve as the template on which the complementary strands is built. So this method of DNA replication is described as the semi-conservative method.